you need to understand that prior to the phase 1 study, if you have a mammalian cell-based product, you need to have data about a viral clearance. So you need to do initial viral clearance studies in order to support the use of that cell line, which again, it is expensive, it takes time, and people are not so willing to do that at that point of time, but that's a regulatory requirement. The second requirement is that if you manufacture an aseptic process, which is usually the case for biologics, then you need to conduct, based on European regulation, 3 aseptic process simulations in order to ensure that you have aseptic process that will result in sterile product.
Are you preparing your IND filing and wondering when you should start and what should be included? Or maybe you're just starting your biological development journey and want to avoid costly mistakes down the road? Welcome to the Smart Biotech Scientist podcast. I'm David Brühlmann, and today we are joined by Rivka Zaibel, who is the founder and president of ADRES Advanced Regulatory Services. With over 36 years in biopharmaceuticals, including 20 years dedicated to regulatory and quality management of drugs, biologics, medical devices, and combination products, Rivka is our guide through the regulatory maze of biologics development. Whether you're commuting to work feeling the pressure of your CMC development responsibilities or heading home looking for solutions, this episode delivers actionable insights on the regulatory and CMC strategy. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Rivka. It's great to have you on today.
Thank you, David. It's a pleasure being here today. Thank you for inviting me.
Rivka, share something that you believe about bioprocess development that most people disagree with.
Okay, so I thought about that a lot. I think that today there is more consensus about the bioprocess development. But like 40 years ago, when things just started and the first recombinant DNA products were approved, there was people actually didn't understand the bioprocess, and the regulators were very strict about that. The process is the product is the process. If you change anything in the process, then you will get a different product. Today, there is an understanding about the processes, and also there is not only one bioprocess. There are so many different biological products today that you cannot talk about one. The way that you manufacture a recombinant DNA product, the protein is not the same as cell therapy, gene therapy. So there are some similarities, but there are differences. And the most important thing is to understand the process for each product so that it makes that more easy and to develop that and to get to a product at the end of the day.
Yeah, I like that you're saying it's important to understand the process. That's so key. And before we dive more into the regulatory aspects, let's talk first about yourself, Rivka, because looking back at your 36-year journey in the biopharmaceutical industry, what initially drew you to this field, and what were the defining moments that shaped your path to becoming a regulatory expert and founding your own company?
Okay, so my story is an interesting one. I actually started as a secretary at a biotech facility 36 years ago. I loved biologics and chemistry since I was a child, but I never had the option to get the proper education. And then, after 2 years of working in the facility, I realized that I am really good at that, meaning assisting my boss in his job and understanding things. And after 5 years of working at the facility, I was given the option of set up a regulatory department, which I was the head of that department until I left the company after 21 years. And during that period, I learned everything about regulatory and quality aspects. It was the best school that you could have had. It was a small company with different products. So I actually was involved in all the aspects of development, from the idea to having the product in the market, from biological processes to aseptic processes. So after 21 years, I realized that in Israel there are a lot of startup companies, biological, which they don't have the proper support. Most of the consultants at that time were from the drug industry, chemistry. There were no really biological consultants that could help those companies. And one of the things that I realized is that they get grants for 1 or 2 years and they don't know what to do with that money. So sometimes they wasted the money in things that are not relevant for that stage of development. So they came to the stage of being able to go to a phase 1 study, but they didn't have more resources. And when I was thinking about that, my idea was that my new mission will be to educate them, to train them, to explain them what is important at this stage and what is less important, what is required from a regulatory perspective and what can be postponed to a future step. And that's what I did. And I had clients very soon because it was really needed. And I started to bring people to work with me, young people, again, with the idea of training them, help them to think the way that I think, not to be very strict about things, to think out of the box, to find solutions to things that seems not to be easy to resolve. And yeah, that's how I came to where I am today.
That's a fascinating story, Rivka. Thank you for sharing that. Now, as a startup founder or as a C-level biotech leader of a small company, sooner or later, you have to start thinking about your CMC development and the regulatory aspects. So let's start breaking this down. First, by starting a bit, how does the landscape look like today? And then I'm going to take it step by step to then coming to a point, okay, what do we need to do in order to have a solid regulatory strategy? So let's start first with the landscape and how does the biologic world look like today?
Okay, so I think that the biological world today is full of technologies and new incentives. What is that? The Biotechnology industry is well established, but still there are new projects in that area. From one side, you have biotechnology companies that develop new products and new technologies, or that they actually improve the technologies available. And there are companies that develop biosimilars, and there are a lot of them. And there are all those new companies or startups that are in developing cell therapy and gene therapy. That becomes to be a very important area, because thanks to that you can have the personalized medicine and advanced therapies to different diseases. Also, there are companies that are developing now microphages as treatment. So it is a world which is full of new development, and each one of them is a bit different from the other. It's not that you can have one guidance that covers all of them. You need to understand your specific process, the requirements of that process, the environmental requirements which are critical for the development and the manufacture. And one of the things that I believe in is that you need to go and consult with the regulatory authorities very early in the process when you are talking about bioprocess, because there are so many small things that can bring you to a success and a failure. And you need to understand that, and you need to have the right agreement with the regulatory authorities, and not only with one. There are a lot of companies that come and say, I want to go to the FDA, and it's okay. And what I usually say is, that's fine. Let's develop a strategy for FDA, but let's also think what EMA will require, so that when you decide that EMA is relevant for you, you have one process. You don't have two different processes. You don't need to repeat things. You can incorporate all that in your strategy. So that is, I think that the challenge of the world today is that there are so many different technologies, and you need to understand, and it's not just like taking some chemicals and mix them. It is more complex, and there are processes that usually you would like to postpone them because they are very expensive, but in this area you cannot. If you need a cell bank, you need to have that as early as possible. Otherwise, really, you may have two different products if you don't develop that correctly.
Yes, I want to chime in here because Smart Biotech Scientist, this is key. CMC development should not be an afterthought. You should start as quickly as possible and also start interacting with the regulatory agencies very early on so that you know what you need to develop.
That's right.
How should we then go about the CMC strategy, and what would you consider the non-negotiable quality or regulatory considerations for a startup company? Let's say we're developing our asset for phase 1.
So, first of all, there are regulations for phase 1. FDA has a guideline of what is needed for a phase 1 biological submission, and IND for a biological submission. The EMA has guidelines related to that. Today we have all the ICH guidelines, which provide you with good guidance about all the aspects. So, first of all, the first question that people ask me is, for instance, I want to start the toxicity studies as early as possible. Can I use a research cell bank in order to do that? So the answer is, if you are sure that from that research cell bank you are going to manufacture your master cell bank, then it is fine to start your toxicity studies with that research cell bank. But you need to start developing, manufacturing your master cell bank and have the initial data about the stability of the genetic stability, that you have a clone that is unique, it's not a mixture of cells. So that is something that you need to do as early as possible in the process, although it costs a lot of money. And you need to take into account that from that step until you have a GMP material, it might be like 9 to 10 to 12 months. And that is mainly for the recombinant DNA processes and other processes that are based on cells development. And then you need to understand that prior to the phase 1 study, if you have a mammalian cell-based product, you need to have data about a viral clearance. So you need to do initial viral clearance studies in order to support the use of that cell line, which again, it is expensive, it takes time, and people are not so willing to do that at that point of time. But that's a regulatory requirement. The second requirement is that if you manufacture an aseptic process, which is usually the case for biologics, then you need to conduct, based on European regulation, 3 aseptic process simulations in order to ensure that you have aseptic process that will result in sterile product. So those are 2 things that are time consuming, are expensive, and are required prior to any treatment of human people. So it's prior to the first in human studies.
When should you start with that? Would that be ideal to start already while you're still in the preclinics? When do you usually advise founders to start with all these preparations?
So it is recommended that the viral clearance study is conducted with material from the preclinical studies. Otherwise, if you wait until you manufacture the product for your clinical study, it will delay the initiation of the study, because you need to have the results of this study, which is not a full viral clearance study, but with some representative viruses, to understand whether your product is robust enough to eliminate or disactivate them. And the 3 septic process simulation, that's something that you need to do just before you manufacture your GMP material.
Excellent. Thank you for clarifying. And I was cutting you short. So there was a third aspect you wanted to talk about.
I just wanted to talk about the ICH guidelines. And today you can find it in those guidelines mainly for gene and cell therapy, and the FDA guidelines related to that. There are provisions there what is needed for each step of development. It's not like a general guidelines that you need to consider what you think is important at each step of development, but today the FDA guidelines related to gene and cell therapy are very clear about what the FDA expects to see at each step of development. So that is very helpful. And here also there is a difference between FDA and EMA. The European authorities rely on the ICH guidelines, which are not specific to cell and gene therapy, in a lot of aspects of CMC, of manufacturing.
Can you give an example, Rivka? Because yes, you mentioned the US Food and Drug Administration and also the European Medicine Agency and the ICH guidelines. There are differences. Each agency, so the US or the Europe, they have different viewpoints sometimes. What are the main differences we should watch out for?
So, first of all, I would say that it's not major differences, it's differences in the details and the interpretation of the ICH guidelines. So, for instance, if we take the example of the aseptic process simulation, in the European guidelines, it's clearly that you need to conduct 3 APS prior to the first GMP material, before you release a GMP product to a first-in-human study, you need to have 3 consecutive successful APS, aseptic process simulation. FDA in the guideline for the first-in-human study states a media fill, which is the equivalent to APS. So it doesn't say 1, 2, 3, and the interpretation of 1 is also acceptable by the FDA. This is one example. Another example is about the validation of the analytical methods. According to the European regulation, you need to have full validated analytical methods prior to phase 3 for sure, and sometimes some of those also prior to phase 2. The FDA will accept, although they prefer to have all the methods validated prior to phase 3, it's not, if I am not mistaken, written in so many words in the guidelines. So those are differences which are more in the interpretation rather than huge differences between the two agencies. The area which is not necessarily related to CMC but more to preclinical studies, EMA is more tolerant to have less animal studies than the FDA. Sometimes companies required by the FDA to conduct several animal studies, while EMA will reduce that number. So that is a difference. And in the area of cell and gene therapy, for instance, I was looking at the guideline for comparability. FDA developed a specific guideline for comparability related to cell and gene therapy, and that guideline, as I mentioned before, also details what is required for comparability at the different stages of development. EMA says that they work according to the ICH Q5 guideline for comparability. So that is a difference. I think that it can be called a major difference because the requirement of FDA are more detailed and more demanding.
What advice would you give us, founders, or as we're leading a company, where should we go look at? I guess we can find all these documents on the respective websites and then find all the details, or what is your approach there or advice?
So the first source of information, I would go to the ICH guidelines. There is a series of ICH guidelines, Q in the quality, that are specific for biologics. So first of all, go there. There are the requirements for cell banks manufacturing, for cell bank testing, viral clearance studies. All that is there. You can learn a lot from there. And then look at the specific websites of FDA and EMA to see whether the interpretation of each one of them is similar. Sometimes it's the same, sometimes there are differences, as I already mentioned. So that is the order in which I do things. Another source of information is if I am developing a product that is similar to a product that was already approved, then I go to the EMA website and look for the EPAR, the summary, the document that summarizes the EMA discussion about the product and the approval of the product. You can find there what were the requirements for that approved product, and you can learn from that. Even if it's not the same product, but if it's the same technology, you can learn from that what it is expected from you. And the same in the FDA, there is summary basis of approval of each one of the products approved. So you can get them under freedom of information and understand what was required for those products.
That wraps up part 1 with Rivka Zaibel, who is the president and founder of ADRES. We've uncovered critical insights on the regulatory requirements and CMC strategies that could transform your development process. If you're tackling complex CMC development questions, I offer you a free consultation to evaluate and refine your CMC development strategy. Book your free call at brühlmannconsulting.com. Join us next time for part 2, where Rivka shares further insights on regulatory affairs, meetings with regulatory agencies, and cross-contamination challenges. See you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.